This is NNI Bites Big Learnings, a podcast where fellow healthcare professionals converse about the latest on nutrition science. Join us as Hania Shayevska and Alexandra Zhernakova share their knowledge and insights on the infant gut virome, knowns, unknowns, and avenues for future studies.
So hello and welcome to today's special episodes of the Nestlé Nutrition Institute podcast, NNI Bites Big Learnings. Today we'll be discussing the infant gut virome, knowns, unknowns, and future studies. Well, one of our Belgian colleagues said that we are not just humans, that we are actually walking microbial colonies. And a big part of those microbes include viruses, collectively known as the virome. And understanding the gut virome is really especially crucial for us, for pediatricians, as it plays, as I understand it correctly, a significant role in the development of health and disease. I'm Hania Shayevska, I'm professor and chair of the Department of Pediatrics at the Medical University of Warsaw, Poland, and I will have a privilege to have and to discuss it with Professor Alexandra Zhernakova, Sasha Zhernakova, MD, PhD, full professor of the Human Genome and Exposome. She's with us, and she's an expert on the virome, and we will discuss this very exciting topic. So Alexandra, we are really excited to have you here. But before we start, could you please introduce yourself and to our listeners, and could you tell us what inspired you to specialize in this field, not in microbiota as such, but in the viruses?
Thank you, Hania. Thank you very much for your kind words. And about myself, I studied medicine. I'm a pediatrician by education. I worked as...
You are also a pediatrician?
I'm a pediatrician, yes.
Excellent.
I worked as clinical geneticist. Actually, I did my specialization in genetics, and then I was doing my PhD in genetics. Then slightly we start making the analysis of microbes using genetic methods.
And
that's how I came.
And it has turned out there are also viruses.
Yes. So actually, I would not call myself an expert in viruses. I don't think I'm expert in particularly virome. We are studying microbiome a lot, but at a certain moment we thought, okay, we have equal number of viruses as microbes. We know that they are really very important in mediating the gut ecosystem and mediating the microbial population in general. So why do we miss that part? Okay. Let's go to viruses, and then we started also analysis of virome in our datasets.
So you already started answering my question, which I have here. How you explain to the regular pediatrician, standard pediatrician, what is the infant gut virome and why it is so important? You already started to talk about it.
So I think you also already mentioned infant gut virome is just a community of all viruses that we have in the gut. We do have a lot of viruses. It's more or less the same as the number of bacteria in the gut. So actually trillions. It's equal, the number of bacteria and viruses.
And is it true that 90% of the viruses are identified as bacteriophages?
Yes, that is also true. So actually it's more, it's about 99%. Okay. We think. We don't know majority of viruses. It's really a dark matter of the gut.
But could you explain to the audience this term bacteriophages?
Yes, of course. So we do have viruses of humans. Of course, we know a lot of them because we know them for their pathogenic properties, for example, herpes virus, enterovirus, and so on and so on. But of course, there are also viruses of bacteria, so they do not infect human cells, they infect bacterial cells, and by that they are called bacteriophages. Okay. There are also viruses of archaea and all other communities in the gut.
Okay, and can you tell us what are some key findings, recent findings about the infant gut virome? Because let's say a few years ago we did not hear so much about gut virome. Now, when you go to the meetings, you hear this term again and again.
That is true. I think this field is still in very infancy. So we just really started scratching the surface of understanding of the gut.
Like with microbiota, I would say.
Yeah, but I think microbiota is years ahead. Years ahead. And of course, with the viruses, the problem is its complexity. We do have a lot of viruses, but they are much, much more individual specific than human microbiome. And by that, when we are studying new cohorts, we identify new viruses. Most of these viruses are actually never been described before. So we do not know them, we do not know their names, we do not know their properties. We have to characterize it from scratch.
So for every infant, it's a different kind of gut virome.
Yes.
It's a different kind of gut microbiota, including or specifically gut virome.
So if you would look for bacteria, I think you would appreciate that every person is different, but at least there are bacteria that are common. Yes, it's not so many of them actually. The common microbiome includes maybe 10, 15 bacteria. The rest will be still individual specific, but you would have sufficient number of bacteria that almost every baby will have.
It's even more
with
complex.
viruses, it's at least times more diverse. That means times more individual specific. And that, of course, makes it so. One challenge is that we do not know much. We have to characterize them. But the other challenge is that all our studies are quite underpowered to see the difference in the gut virome. So on the individual level, for one particular virus, we probably can see how it comes from mother to baby. We do see effect of breastfeeding, for example, on the virome composition, on diversity, on some viruses.
You see the effect of breastfeeding?
Yes, we do see that, for example, babies who receive breastfeeding, they have higher diversity of viruses, babies that receive formula feeding, they have higher diversity of viruses than babies that receive breastfeeding. That's what we see in our data. But again, for majority of phenotypes, for majority of health factors, medication, for example, we do not have sufficient numbers to make conclusions of viruses because there are so
And we're saying
diverse.
Sufficient samples. So what do you mean? It's like hundreds, thousands?
I think for the low-hanging fruits, we probably will be there with thousands, but for the rare situations, such as individual variations in medications or rare effect during pregnancies, there we need larger numbers. But of course, it doesn't say that viruses are not important. Yes, if you would find some viruses that mediate the abundance of bacteria or that transfer certain antibiotic resistance genes, for example, then we need to study that and we need to follow these examples.
You're a pediatrician, so you probably understand very well that pediatricians would like to know what are the potential clinical implications of better understanding of the gut microbiome, because it's nice to know it, but what are the practicalities of such knowledge?
Yes, I think that there are two major things that are worth discussing. One, of course, we know that viruses can mediate abundance of bacteria. They can be treated as an alternative of antibiotics, and antibiotic resistance is something that is warning. So there is a development of looking for the virome cocktail, bacteriophage cocktails that can treat diseases.
It used to be quite popular in Eastern Europe.
Yes, it is still. It still is popular. Yes.
But now becomes also like
yes, there are some very pioneering study. For example, it was a paper in Cell a few years ago by Rana Linaf group in collaboration with Boston, with MIT, where they developed the cocktail of viruses that can protect against Klebsiella, pathogenic Klebsiella, that is associated to inflammatory bowel disease, for example. I think it was a very extensive study. It is kind of a pioneering study in this field. Still, on the human level, they assessed that it is safe, but the study mainly addressed the inflammation in the mouse models. But I think it is a very promising and very nice study. So one development of virome is treatment. Another thing that is very interesting are some studies, for example, from Colin Hill group, where they looked for the virome microbiota transplantation, so not fecal, but the fraction without bacteria. And what they could see that indeed this virome fraction can initiate the phenotype, just like FMT
But these are animal
does.
studies.
It's animal studies, but I think that means that viruses
Well, but it's
mean
important,
something.
it's to understand
Yes.
the animal, not human studies as yet.
Yes, it's not yet there, but I guess that this actually brings another very important aspect. When we are making the FMT, we are making them in humans, but I think the question of safety is actually something that should be very properly addressed there. And we tend to forget that it's not only bacteria that can mediate our disease, but maybe also cause disease. And there are also virome fraction.
But as you said, one of the problems that we have with FMT, fecal microbiota transplantation or transfer, is that we don't know what really is active ingredient.
Exactly, and many companies are trying to make the combination of bacteria, and I think it was very promising. But as we see now, it doesn't work as magic, at least. I would not say it doesn't work at all, but it's not really magical treatment of any diseases. Maybe because we need also viruses to be there, and of course metabolites and other things.
Well, some of our listeners are probably young researchers, and for young researchers studying the infant gut virome, so what should be, in your opinion, the top priority? And if it's really, would you say, this is something to study? Definitely the answer is yes, because this is what you are doing.
Yes.
And what are the major problems I would say, like methodological, because there must be some methodological problems.
There are plenty, yes. Yes, there are a lot of methodological problems. One problem that we do not really know what we are studying, so it makes it all very challenging. On one side, the lab protocols need optimization for that, so it's also a constant process of optimizing and making the best protocol for that. Secondly, the bioinformatic pipelines, there is no really existing golden standard pipeline, so you have to develop that. It's really a lot of pioneering work there. So that are, I think, biggest challenges for the virome field. Now, yes, I think that there are a lot of exciting and completely unknown things in the viruses. For example, one thing that I find particularly exciting is the interaction between viruses and bacteria. They are very dynamic. They are very complex. For example, bacteria have an immune system against viruses, and it's not only CRISPR that we all at least know, but there are multiple clusters of genes that bacteria are developing to protect themselves against viruses. But there are also viruses that catch some bacterial genes to their genome to make sure that they have evolutionary adaptation. There is transmission of antibiotic resistance genes, of other genes, the mobilome, so the development of gut ecosystem is really coming as an interaction of bacteria and viruses. And I think we do not know much there, and for young researchers, it's plenty of opportunities to study.
And now everyone talks about artificial intelligence and machine learning. So is it something that you are already applying in your lab? Or not yet?
We are doing prediction methods, but I think that we still do not use these sufficiently. So I'm very much looking forward to our further discussions on that. But I think this is something that will be very, very, very helpful for the virome field.
Okay, so you encourage young physicians.
I encourage young people to study artificial intelligence.
So thank you, Alexandra, for sharing your insights on gut virome. It's really something that we are discussing, as I said, again and again. So gut bacteria is not just about just microbiota. It's not just about bacteria. It's also fungi. Also, we did not discuss it.
Yes,
but this is another interesting topic and research area, but it's also about viruses. As we said, 90, and you said 99% of the viruses are identified in the gut, which are identified in the gut are bacteriophages. Yes. And they target intestinal bacteria, if I understand correctly, not human cells. So this is important.
Not human cells.
Exactly. So they do not pose any threat to children, which I think it's also important to understand.
Yes, and I think that's actually a beauty. We also have viruses, of course, everywhere. Yes, also in breast milk, for example, there is also a virome. And we know that this is safe because children are drinking breast milk. So this is a big potential of looking for something that is for sure safe, but also can mediate the microbiome.
Thank you very much. It has been a pleasure, and I do hope we will have occasion to talk about it more, because the research is, developing, evolving, and I'm sure we will hear much more. Thank you very much, and thank you to our listeners for joining us today. Stay tuned for more discussions on the latest in health and nutrition research. Thank you very much.
Thank you.
And that concludes today's episode. Thank you for joining us with Hania Shayevska and Alexandra Zhernakova as we talked about the infant gut virome. knowns, unknowns, and avenues for future studies. We hope you enjoyed and learned from this quick discussion, fellow experts. Till the next episode.